Okay, hello class. Welcome to our post lab. And today we have bacteria as part one, and then for part two on Friday, I think we're going to discuss protozoan and fungi. Okay, so this is a blood agar. So instead of a regular agar plate. So this blood agar is a bacterial growth medium also, but it's kind of enriched. Okay. So why do we need to use enriched bacterial agar plate? So sometimes we have fastidious organism that require other nutrients, okay, such as streptococci. And blood agar is usually made up of trypticase soy agar, and this is enriched with 5% sheep blood. So there's blood, that's why the color is red. Okay. And of course, the blood agar encourage the growth of some bacteria such as streptococci. And take note also that some bacteria cannot be grown using blood agar plates, and those bacteria actually belong to Neisseria group and Haemophilus group. So in order to grow them, we could heat the blood agar, um, and then after we heat the blood agar, we inactivate some of the enzyme. And heating the blood agar, then we produce chocolate agar. Okay, it's color brown. That's why they call it chocolate agar. And take note, mm, yeah. So, so the blood agar, we have um er, blood cells. Okay, we have the hemoglobin, and take note, we could also characterize the bacteria or distinguish the colonies on our blood agar plates, whether they are hemolytic bacteria or non-hemolytic bacteria. So here we have alpha, beta, and gamma hemolytic bacteria. So what are the different types of um, hemolysis? So if it's an alpha hemolysis, take note the color. It seems like greenish. So this colony, they are capable of um, degrading the hemoglobin, but not much. So that's why, like here at this portion, there is a reduced number of hemoglobin. So there is a like a partial uh, hemolysis. Okay, but beta, there's a very clear zone, so it can to like complete uh, hemolysis of the hemoglobin, while the gamma doesn't have hemolytic activity. Okay, so in this discussion, we will use blood agar plates to describe the colonies of uh, the bacteria. Okay. Okay, first is of course we have here Streptococcus pneumoniae, and sometimes it's misleading, but Streptococcus is not chain-like, but actually the arrangement is Diptococci, like in pairs. Okay, and there it's gram-positive, and take note the clinical relevance bacteria causing pneumonia. And it could be also associated with ear infection. Of course, when it goes to blood, it can also cause sepsis. And take note, this is sputum, and you can get sputum and check if pneumococci or pneumococcus pneumoniae, streptococcus pneumoniae, sorry, are present in the sputum by gram staining. Of course, under like on blood agar plates, like the colonies appear like this, and it's round and glistening and develop a dimple, then a crater-like appearance. We call this morphology or appearance as Dropsman colonies. Okay, so take note, Dropman's colonies could be flattened or concentric ring with depressed center or central umbonate. Sometimes there is like peak here and then depression here. Okay, peak at the center, then depression, and then here is um like normal. Okay, so remember there's a depression because the um bacteria is kind of hemolytic. Okay, due to the hemolytic activity. Okay, so take note. So if you get a sputum, collect sputum, then try to um, inoculate or spread some sputum on blood agar, and you see this kind of colony. So you could um, infer that the person has pneumonia, okay, or Streptococcus pneumoniae. And don't forget, Streptococcus pneumoniae is a alpha hemolytic bacteria. So as you can see, it's not so clear, like a little bit. So it's alpha hemolytic bacteria. Okay. Another diplococci. Of course, we have Neisseria meningitidis, but this time instead of compared to gram-positive Streptococcus pneumoniae, we have here a gram-negative bacteria. And from the word meningitidis, so of course the it can cause meningitis. Okay. And another form of meningococcal disease, such as meningococcemia. So meningo, like meninges, like the membrane um, found in our brain, and coccal, of course, cocci, like ground bacteria in the meninges. Okay. And of course, in the bloodstream, when bacteria goes in the blood, it can cause sepsis. Okay. And under on blood agar plate, they are non-hemolytic bacteria. Yeah, don't forget meningo, meninges of the brain, and meningococcal. Okay. Another take note, this is Neisseria species, but it could grow on blood agar. And as I told you, some bacterial species, they cannot grow on blood agar like Neisseria gonorrhoeae. So Neisseria gonorrhoeae cannot grow on blood agar, so we use chocolate agar plate. And from the word gonorrhoeae, of course, it can cause gonococcal infection. Gono means the gonads or the reproductive tract infection. So, example, we have the STD, so gonorrhea. Okay. So same, Neisseria. So it's a gram negative and diplococci, so Neisseria gonorrhoeae. Like the most common bacteria found on our skin, and we have different strain uh, of Staphylococcus aureus. So from the word staphylo, it's grape-like in cluster, and coccus round shape. And this Staphylococcus aureus is a gram-positive bacteria. So here we have beta hemolytic Staphylococcus aureus, and some Staphylococcus aureus, like this methicillin resistant uh, Staphylococcus aureus, they are not uh, hemolytic. So take note, class, sometimes the strain of the bacteria, their colonies, colony morphology sometimes change, and as well as their biochemical um, properties. So this is the gram reaction of Staphylococcus aureus, so grape-like, gram-positive, and again depending on the strain, sometimes they appear golden. That's why aureus, like gold, yellow color. Some strains, of course, like they lost pigmentation, like they kind of produce this white colonies. Again, some strain they are hemolytic, others are not, like just alpha hemolytic. Um, and here, sometimes they lost their pigmentation through time. Okay. Sometimes, um, when the colony is too old for Staphylococcus aureus, they begin to develop more pigmented colonies. Okay. Also, take note that Staphylococcus aureus has long been recognized as one of the most important bacteria that cause disease in humans. So, it is a leading cause, of course, of skin and soft tissue infection. Of course, you have abscess like boils or yung pigsa. Then you have your furuncle, carbuncle, cellulitis, yung pimples or yung acne. So when S. aureus goes inside the skin, it can result to a lot of skin infection. Yeah, and another thing, if it goes to bloodstream, of course, it's a serious infection. It can also go result to pneumonia and goes to your bone or joints, so causing joint infection. Okay, but normally inside in it's also part of our skin normal flora. Usually they are not dangerous unless if you're immunocompromised or it happens to be that it goes inside your body, it reach the bloodstream. Okay. And of course, here we have Escherichia coli, Escherichia coli, or E. coli. So here we have gram-negative rod shape or bacilli, and E. coli, they are actually found in low intestine. Most of the E. coli in the intestine, they are not considered harmful. So some of them are actually important for us to produce vitamins such as vitamin K. And you know, vitamin K is important for normal blood clotting. That's why when you take too much antibiotic and kill some important bacteria for vitamin K production. So sometimes you could develop um, what's that? I forgot the term. Can develop um, hemophilia or abnormal blood clotting due to too much intake of antibiotics. Okay, why? Because again, it kills the important microbes in the gut that's responsible for making vitamin K. Okay, and of course, those are the good E. coli. So of course, there are also some strains of E. coli which are considered harmful, and since those pathogenic E. coli, of course, can cause is associated with uh, diseases of our GI tract. Okay, so yeah, so under like on um, blood agar plates, it appears like um, this, and they are actually not hemolytic bacteria. So it's a gamma hemolytic bacteria. So E. coli is a gamma hemolytic bacteria. Okay, next we have Pseudomonas aeruginosa. Um, so Pseudomonas aeruginosa, it's a gram negative here. So it's also rod, gram negative rods, and they have polar flagella. And the thing is, they are known as Piocyanine or blue-green, uh, Pioverdine or yellow-green, or Piorubin or red-brown producing strain. Okay, so it depends to the strain. So sometimes they have color, and sometimes, usually when we grow bacteria in the lab, like pigmented bacteria, eventually they lose their ability to produce color once we grow them in this kind of media or like the artificial media. Okay. And take note, Pseudomonas aeruginosa, like it is commonly found free living in moist environment, but it's also a pathogen of plants, animal, and of course, human. And although they are aerobic, although an aerobic atmosphere is necessary for its optimal growth, like most strain can multiply slowly in anaerobic environment. So if the nitrate is present as a hydrogen acceptor. Okay. Okay, here we have Klebsiella pneumoniae, and of course Klebsiella pneumoniae from the word pneumonia, and it of course can cause pneumonia associated with different kind of infection. Of course, you have blood infection, wound, surgical site infection, and if it goes to your brain, of course, it can also cause meningitis. Okay, so under microscope, after gram staining, this is the appearance. It's a rod-shaped bacterium, and it's a gram-negative bacterium. Um, yeah, on blood agar plate, it doesn't have lysis pattern, so it's a non-hemolytic bacteria or a gamma hemolytic bacteria. Okay. Okay. So next we have Mycobacterium tuberculosis. The media for this is quite um, different. Here we have Lowenstein Jensen medium, this greenish color, bluish color. media, and under the on this media, uh, look at the appearance of the colony. Okay, it's quite different, and it doesn't react well with gram stain. Okay, I already explained this in the previous topic, like because of the high presence of mycolic acid, and instead we use acid fast staining. Okay, so from the word Mycobacterium tuberculosis, so of course it is associated with tuberculosis. Okay.